Welcome for another episode on Colony PCR with me, Rex. So, let me first explain what is a colony PCR. So, when you do cloning, you do transformation, you get multiple colonies, and you want to screen for the colony which contains your gene of interest. So, what you do actually is we try to pick up the colony and set up a PCR to check whether the gene of interest is cloned in that particular E. coli cell. So this is called as colony PCR, where we use the colony as the template and not isolate any plasmid or DNA. Once we screen, then we can isolate the DNA, plasmid DNA. So assume that the DNA is cloned or the gene of interest is cloned inside the plasmid. There are two options. You have a gene specific primer and you also have a primer sequence on the flanking the cloning site in any cloning vector. So you can use either the gene specific primers or the primers that are available on the backbone of the vector to amplify. So colony PCR exactly is you just inoculate the colony in the PCR tube containing the PCR reaction, and the denaturation temperature will break the cell open and the DNA will be used as a template for amplification. So the primers will bind to the denaturing DNA, the plasmid DNA which you have transformed, and it will amplify the fragment if it is present. So this is what we are going to do today. And we are going to run it on a gel and we are going to see what the results are. This particular colony PCR helps when you screen a very large number of transformants or recombinants. You can pool the recombinants even. Say 5 colonies can be picked together, inoculated in one reaction, and if any one is positive, any one out of that 5 is positive, then we can separately analyze. This is the reaction setup. So how do I set up a reaction? Say we are going to analyze 5 colonies today. So what we are doing is we have listed the requirements: sterile water, 10x reaction buffer, primer 1, primer 2, that's forward primer, reverse primer, NTPs which is 10 millimolar, Taq polymerase, and template. So the first thing that you see is the Taq polymerase, which is 3 units per microliter, and we have 10 microliter with us. So we can divide it in such a way that we can add 1 unit per reaction. The second is the buffer, which is a 10x buffer. The buffer contains magnesium chloride, final concentration 1.5 millimolar. The third are the primers. The primers, there are 2 sets of primers. One is the flanking primer, one is the forward primer, the other is the reverse primer. In our case today, we are using M13 general forward primer and M13 general reverse primer. So these primers, they lie on the flanking backbone of the vector, PUC vector. So we don't have to use a gene specific primer. This is followed by the DNTPs. The DNTPs are 10 millimolar DNTPs, 10 millimolar each, ACTP, GCTP, CCTP, and TTP. So all the 4 nucleotides are mixed and the final concentration will be 10 millimolar each. So what we try to do, we try to mix all these contents. So the template DNA will be donated by the colony. So we prepare the mix, master mix for 5 reactions. We usually prepare a cocktail when we prepare more than 1 reaction, and we aliquot into individual tubes. So we try to aliquot all the reactions into individual tubes. So we have taken 5 tubes plus 2. The plus 2 is, one is a positive control, the other is a negative control, and 5 tubes for the colonies. What is a positive control? A positive control is a control that we use where the reaction will not fail, which means that that positive control template is a plasmid DNA, not a colony. So we will be so sure that your plasmid is amplified. These are the colonies that we are going to pick today. So we have 5 colonies from the transformed plate. So I am choosing the 5 colonies, 1, 2, 3, and I have already circled the colonies so that we can pick and use them. So there are 2 ways of doing it. One, we can patch the plate and then allow it to grow overnight, and then from the patched or grown plates, you can set up a PCR. Again, using a PCR, you can directly inoculate the colony, or you can resuspend the colonies in 10 or 20 microliter of sterile water and then use it. Right? However, today I am going to use the colonies straight. So the positive control and the negative control do not have any template. The positive control will have the plasmid as the template, and the negative control will go without template. It is a non-template control. So I have two plates with me. One is a fresh LB agar. The second is a LB agar which is transformed, which has got the transformed colonies. So I'll use a tip. I'll use a tip and pick up the colonies from the transformed plate, and I will patch on the freshly prepared LB plate with the selection. And then I will use the same tip and mix it with the PCR mix that I have already prepared. Just swirling a couple of times is sufficient to dislodge a few colonies. That will be more than sufficient for the PCR. So we have 5 colonies, and I am patching all the 5 colonies and also inoculating them into the PCR mix. So this is time efficient, because once you set up the PCR, you will know that how many of them are positive, and the same evening you can inoculate the positive clones for plasmid isolation for the very next day. So this speeds up the process, but some people will always patch, then use the grown colonies for PCR. The other way around is they pick the colony, inoculate it in water, use 2 or 3 microliter of that colony load in water as the template. Since I am comfortable with this, I use this method of colony PCR. So I will store both the plates in 37, and then I will mix the tubes. I will mix the content of the tubes using a spinner. So this is to ensure that the colonies and the PCR mix is completely mixed, one, and the second one is there should not be any content on the lid or on the walls of the PCR tube. So I spin it nicely, bring all the contents down before I set the PCR reaction. So when I set the PCR reaction, I will have to set the conditions for the PCR reaction. The most important condition for PCR reaction is the melting temperature. So the melting temperature that we use, or the annealing temperature that we use, is 55. The denaturation temperature is 94 degrees Celsius, and the extension is 72 degrees Celsius. So the rule of thumb, as a rule of thumb, we use 1 minute per kb, and if you are amplifying 1 kilobase per fragment, we use 1 minute for extension. So once we set the PCR reaction, we confirm that all the program is, whether the program is correct and all are good, and then we run the program. Once we run the program, we have time to cast the gel, and we will prepare the gel so that once the PCR is over, we can load it onto the agarose gel. Here we use 1% agarose gel, and we have loaded the sample. This will be run at 100 volts for 10 minutes using TBE buffer 0.5x strength and visualized under ultraviolet light. So for our easy convenience, I have used a picture of the same gel at the right bottom corner. The orange fluorescence is not so clear because the lights were on for taking the video. So I have inserted the black and white picture of the same gel. The first lane is the positive control, where the plasmid was used as the control. The second is the negative control, which is NTC, non-template control. The third, fourth, fifth, sixth are the colonies. So you can see that the third colony is not amplified. The third colony is a negative. The first, second, fourth colonies are positive. So we can pick the colonies inoculated for the next day's plasmid isolation. Thank you for joining me. Bye-bye.